Okay, well, here we go. So I want to go over this article, and then we'll go a little bit into more detail as far as what exactly happened. So a year in, the first patient to get gene editing for sickle cell disease is now thriving, which is amazing. So I'll only go over the first few paragraphs. So like millions of other Americans, Victoria Gray has been sheltering at home with her children as the U.S. struggles through a deadly pandemic and as protests over police violence have erupted across the country. But Gray is not like any other American. She is the first person with a genetic disorder to get treated in the United States with the revolutionary gene editing technique called CRISPR. So, and as the 1 year anniversary of her landmark treatment approaches, Gray has just received good news that the billions of genetically modified cells doctors infused into her body clearly appear to be alleviating virtually all of the complications of her disorder, which is sickle cell disease. So here, it sounds like I didn't go too far into the detail of her particular procedure, but it sounds like they have extracted some of the red blood cell producing bone marrow tissue out of her and did some genetic engineering on that bone tissue using CRISPR, and then re-injected those back into her to produce healthy red blood cells. Granted, I don't know exactly if that was the case, but that would be my presumption. So what is sickle cell disease? So sickle cell disease is a single genetic disorder which causes hemoglobin in the body to basically crystallize, and it forms long rods of hemoglobin, which then interrupt the normal red blood cells' ability to have this nice donut-looking shape. So the cause of it is genetic, and let's go down. Okay, so normally humans have hemoglobin A as the predominant or most of the hemoglobin in the adult human body is of the form hemoglobin A. And this consists of two alpha chains and two beta chains. So if we were to show you a molecule of hemoglobin, you can see here, I don't know which one is alpha and which one's beta, but it has two of alpha subunits and two of beta subunits. And I'm just, guessing right now. I'm assuming, yeah, I won't be able to tell you which one it is right now. So they will call it alpha sub 2, beta sub 2. And so this is the normal structure of hemoglobin. And in people with sickle cell anemia, the two beta chains, and let's try to, I'll just keep on going. So normally in a healthy person, hemoglobin A consists of two alpha and two beta chains. There's also hemoglobin A2, which consists of two alpha and two delta chains, and hemoglobin F, and I assume F stands for fetal, and this consists of two alpha chains and two gamma side chains. So here you can see with hemoglobin, frequently we are actually using the same alpha chain, and then it's acting as a module with which we can change the other modular part, such as the beta side chains or the delta side chains, or delta chains, beta chains, or the what's it called, the gamma chains. So let's continue. In people diagnosed with sickle cell disease, at least one of the beta globulin subunits in hemoglobin A is replaced with what is known as hemoglobin S. So it is an S subunit standing for sickle cell, I would assume. So it's an S subunit instead of the typical beta subunit that you normally see in hemoglobin A. In sickle cell anemia, a common form of sickle cell disease, hemoglobin S replaces both beta globulin subunits in the hemoglobin. So we should skip down to here. So the gene defect is a single nucleotide mutation, also known as an SNP. And so this is, just know that this is the DNA code. GAG gets accidentally changed into GTG, and that GAG would normally encode for glutamic acid amino acid residue. But since it got changed to GTG, it then encodes for valine, which is a different amino acid residue. So glutamic acid is a negatively charged or acidic amino acid, and it's getting replaced with valine, which is a hydrophobic amino acid. And so if you can imagine, in a protein, you have hundreds of amino acids that are all polymerized into one subunit. Well, out of those hundreds, it just took one of those amino acids to turn from their normal acidic residue to a hydrophobic residue, and that was enough. That one mutation was enough to change one amino acid to now allow hemoglobin to crystallize into these long rods. So let's see. What these people decided to do was undo that process somehow, at least in a subset of cells, if not as many cells as possible. So gene therapy has really made great strides ever since the introduction of CRISPR technology, and CRISPR stands for clustered regularly interspaced short palindromic repeats. So I think the best way to describe this to you is through how we discovered this in the first place. So normally, you've probably seen these viruses known as bacteriophages. So this is the stereotypical bacteriophage structure that we see in cartoons and science articles, microscopy all the time. So what this bacteriophage does is it injects its DNA into a bacterium or into a prokaryote in order to take over its machinery to produce more viruses. And that's how it would normally work. However, bacteria have their own version of an immune system against viruses. So the way that this works is if random DNA gets into the cell, there is known as CRISPR-associated proteins, or Cases, that find the DNA, and then they use that DNA to create a novel, what is known as a novel spacer. And this spacer will actually get inserted into the CRISPR array, which is one of the genes within the bacterium. And so this CRISPR array then has, I wouldn't be able to tell you if it's a bunch of different novel spacers or if it's the same spacer repeated over and over again. Either way, it is able to insert this small bit of DNA into this array. And then when this array gets transcribed from DNA into RNA, it ends up producing a bunch of these spacers with these palindromic sequences. So a palindrome is something that that if you read it forward and backward, it sounds out or spells the same thing. So you have these spacers that are in between these palindromic sequences. So then what happens is CRISPR-associated protein 2 will then come to this transcribed section and split it up into these processed, I assume these are short for CRISPR RNAs. And these CRISPR RNAs, or cRNAs, then get put onto CRISPR-associated protein 3. And then this CRISPR-associated protein 3, since this little novel spacer, came from the original DNA. So this teaches Cas3 what DNA it's supposed to interact with. And then when it interacts with that DNA, it then breaks it up. It cleaves it. And so what we have done, quite remarkably, is we've developed what is known as CRISPR-associated protein 9, or Cas9, which we've done a number of different genetic modifications to it to do a bunch of different functions. But one of the things that we have done with it is we have made it in a way, we've mutated it in a way, or engineered it in a way to allow us to create our own processed cRNAs. So this would be an RNA that would target the DNA sequence that you want to target, and then it interacts with our Cas9. So not this Cas, it'll be an engineered Cas, and then that will then interact with that DNA, and then we can cause some form of chemical reaction at that place, and that chemical reaction can usually be conducive for causing genetic engineering in some form. So let's I should go over it a little bit. So is a family of DNA sequences so I should say CRISPR is a family of DNA sequences found in the genomes of prokaryotic organisms such as bacteria and archaea, and these sequences are derived from DNA fragments of bacteriophages that had previously infected the prokaryote. So it sounds, yeah, it sounds like there's one gene and then it gets inserted with new DNA sequences as the bacteria ages. Anyways, these are used to detect and destroy DNA from similar bacteriophages during subsequent infections. So it is a long-lasting immunity. So even, let's say, a different bacteriophage does infect your bacteria, if it has, it might be a different species, but it might still have the same spacer in the DNA of the CRISPR array. And so what happens then is, even though it's a completely different virus, since it has the same DNA, it becomes vulnerable to being destroyed by this CRISPR-associated protein immune system. So, and then let's see. So Cas9, or CRISPR-associated protein 9, is an enzyme that uses CRISPR sequences as a guide. So it uses these CRISPR sequences to guide it to a target DNA sequence in your cell. And it can cleave specific strands of DNA that are complementary to the CRISPR sequence. So Cas9 enzyme together with CRISPR sequence form the basis of a technology known as CRISPR-Cas9, and this can be used to edit genes within organisms. This editing process has a wide variety of applications, including the basic biological research, development of biotechnology products, and treatment of disease. So we really can't learn about CRISPR unless we know about Cas9. So I should say, oh man, this is going to be a little bit too much. So basically, this is what the Cas9 system does. So you have your little RNA sequence that you made, and it has your target sequence inside of this RNA. And so this RNA then interacts with Cas9, and this whole structure is then able to interact with DNA. And then this can cause cleavage at these two sites because there are two not reaction centers. What would the word be? There are two active sites that can cause cleavage at these locations. And so after you've caused cleavage at these locations, you can insert other DNA into this cleavage site, usually using some repair mechanism of the human. Granted, there's a million different ways at which you could cause genetic engineering after you've targeted this site. So going back to the people with this single nucleotide polymorphism, or this mutation in their hemoglobin, you could theoretically target those sequences and then engineer hemoglobin to be normal again. So, or at least not have this variant. And granted, I do not know exactly what type of insert of genetic material they gave these cells, but that would be one of the varieties of ways that you could genetically modify someone's bone marrow cells to then produce healthy hemoglobin rather than the sickle cell anemia hemoglobin. So with that, I think that is plenty of information. So with that, I'm going to get off, but thanks for watching, and I hope to see you tomorrow.